Hello, we are discussing about the gram staining and myself Dr.Kamthane DC, Head Department of Microbiology, SGP College Purna. Gram staining principle we have to discuss about this. This gram staining, it is based upon the differentiation in between the gram positive and gram negative organism cell wall. These gram positive organisms, these are having the peptidoglycan more as compared to the gram negative organisms, while in case of the gram negative bacteria, the lipid it is more as compared to the gram positive bacteria. Here the primary stain used, it is 1% crystal violet, the mordant it is the gram iodine, and the decolorizing agent this is 95% alcohol or acetone may be used, and the counter stain which is used, this is the safranin. First, the application of the primary stain to the bacteria it is there, and this primary stain, that is the crystal violet, when it is flooded, then it takes in both the gram positive and gram negative bacteria. The mordant used, it is the gram iodine, and this is forming the CBI complex in both of the gram positive and gram negative bacteria. Next, it is followed by the decolorizing agent, that is the 95% alcohol. This decolorizes the gram negative bacteria and not the gram positive bacteria. This gram negative bacterial cell wall having the lipid, and it dissolved due to the action of this alcohol, and when the safranin, that is the counter stain, it is applied, it gives the stain to the gram negative bacteria only, and this is the difference between the cell wall of gram positive and gram negative bacteria. And the examples of the gram positive bacteria and the gram negative bacteria. The gram positive organism, these are of two types, cocci and rods. In case of the cocci, Staphylococcus, Streptococcus, Enterococcus, then Listeria, Corynebacterium, Nocardia, Bacillus, Clostridium, these are the examples. While in case of the gram negative bacteria, cocci, rods are there, and spirochetes are also present in case of the gram negative bacteria. And in case of the cocci, Neisseria, Pseudomonas, Salmonella, Shigella, Klebsiella, Proteus, Escherichia, these are the facultative anaerobic organisms, while in case of the anaerobic rods, the bacteroids, Fusobacterium, these are the examples, while the microaerophilic gram negative rods, these are the Campylobacter. In case of the spirochetes, aerobic, these are the Leptospira, and anaerobic Borrelia and the Treponema. And the procedure of this gram staining, it is first prepare a smear on a grease free slide, then this is heat fixed. After heat fixing, the crystal violet it is applied for 2 minutes. After this, the slide is to be washed, and then flood the smear with the grams iodine for 2 minutes, then water wash the slide, then flood the smear with 95% ethanol for 10 to 20 seconds, and see just the stain which is there, it comes out of the smear. Then again water wash the slide and apply the counter stain, that is the safranin, for 2 minutes. After this, wash the slide with the help of the tap water and adjust the flow of the tap water so that only drop by drop water it comes on the smear or slide side and it get washed. After washing the slide, it is air dried and observed under oil immersion objective. In the observation, we observe the gram negative organisms which are stained with the saffron ink and appear as pink, while the gram positive organisms, these appear violet in color and these are differentiated. In this way, the gram positive organisms and the gram negative organisms are differentiated. Thanks a lot for listening.